Hello everyone, welcome to the session Bioethics. Today we are going to discuss about CPCSEA, which is the Committee for the Purpose of Control and Supervision of Experimental Animals, 1964. And this came into existence under the Prevention of Cruelty to Animals Act, that is PCAA, which is Prevention of Cruelty to Animals Act 1960. So what does this committee does, or what is the functions of the committee? So this committee was formed or formulated to foresee that how laboratory animals are maintained, to ensure that the experiments that are conducted were according to internationally accepted ethical norms. The committee has also the power to inspect any place and prohibit any person or institute who is doing an experiment which is not under those internationally accepted ethical norms. So this committee has every right to protect these experimental animals to prevent infliction of any pain or suffering to these experimental animals. So their main function is to see that how they are maintained in an animal house and ensure that the experiments conducted are in the ethical norms. And if they find, or they has the power to inspect any institute or a person or a place who is doing this animal experiment, and if they find that they are doing the experiments which is not under this standard, that is the internationally accepted ethical norms or standard, they can prohibit the person and also can uh, charge that person under this particular act. So the members of who are the members of this committee of the CPCA? The members include renowned scientists, members from the regulatory authorities, and also animal activists. So all labs conduct or who is conducting the animal experiment should be registered with CPCSEA. So a registration from CPCSEA is a mandate for an animal house or an institute who is so mainly animal houses are seen in research institutes or in medical colleges where medical researches happen. In these institutes or research institutes, they should have the particular the institute or the animal house facility should be registered with CPCSEA, because they should follow the guidelines, all the guidelines which is set by CPCSEA, and these committee members will be or they will inspect the place yearly or quarterly to check whether the animal house and also the experiments which is conducted in that particular institute is uh, under the is with the ethically standard or internationally accepted norms or internationally accepted experiments. So coming to the activities of this council, the council is responsible for registration. As we said, they register the institute. So their first activity is to register the institutes or establishment which is meant for breeding the animals, and also for breeding as well as for experiments on animals. The institutes and establishment can be registered, or they should be registered with CPCSEA. Next is approval of animal house facility. So they approve an animal house or register an institute with an animal house only when that animal house is built with the internationally recognized norms. Norms in the sense that, as I said, some of the norms are, one is that when you are having or when you keep different types of animals, they should be kept in different rooms. And because these animals are used for experiments, okay, the animals on which experiments are conducted should be placed in a separate room. They should not be in the stock room. Stock room means where the animals are being kept normally. They will be allowed to breed, raise their number, etc. But in that particular room, which is the normal normal room. That in that room, experimental animals in the sense that the animals on which an experiment is started, or an animal on which an experiment is begun, such animals cannot be or should not be kept with normal animals. Or that means that there should be a separate room for experimentation, or to keep experimental animals, and there should be a separate room for surgery, if surgery is to be done on the animals, if some injections are to be given, if some drug is to be given orally, or whatever experiment is done, an experimenting room should be there which is separate. And another thing is that for animal houses, two corridors will be there. Two corridors in the sense that one corridor will be usually called as a clean corridor, because these animals, or the place where the animals are kept, should always be kept clean. So in the other corridor, the neat corridor or the clean corridor will be in the front, and the dirty corridor will be in the back. Dirty in the sense that always it will be wet, because all throughout the scheduled time, cleaning of animals, then the preparation of animal feed and feeding the animals, etc., will be done in through that particular corridor. As a result, because of the cleaning, etc., that will be always wet or dirty, and then that's why it is called as a dirty corridor. uh, so such simple norms are to be followed, or there are a set of guidelines internationally, or international standardized guidelines for the building of animal house. So only such animal houses will be approved by CPCSEA. Next is permission of a committee for conducting the experiments. So this committee is an internal committee which is made by the institute, which is called as an ethical committee. An animal ethical committee will be there in the institute, and such an institute will be formed by, or will be uh, organized by the guidelines of the CPCSEA. The members, so and so members, etc., that will be followed under the guidelines of CPCSEA, and only this committee can permit which projects should take or should conduct experiments on animals, and which project should get a sanction of animals for experiments, etc. Only the project which is sanctioned by the ethical committee. So ethical committee is an internal committee of the institute. We'll see that in the next slide, an ethical committee or an animal ethics committee. But that ethics committee, even though it is formed by the institute, the members of that particular ethics committee should be formed, or the number of members of such committee should be under the guidelines or pertaining to the guidelines of CPCSE. Next is recommendation for import of experimental animals. So many experimental animals, like the transgenic animals. So transgenic animals are usually they are made usually outside our country. In India, they need to be exported, and after export, they are reared in that particular animal house for their use. So such animals, like transgenic animals means those animals uh, transgenic in the sense that they are not normal animals. For example, you have many transgenic mice. So transgenic in the sense those animals were created only for experiments, only for experimentation purpose. Transgenic in the sense, in those animals, some genes in them, either it will be overexpressed or underexpressed. Okay, there are transgenic mice. For example, for many cancer studies, we use transgenic mice. There are transgenic mice where something which is called as a nude mice. That is a name of a transgenic mice. If you search in the internet, you can get the name. It is called as nude mice, in the sense that mice was made for cancer research, where it is immunocompromised. It does not have any immunity. That means the immune cells are lacking in that animal. And such animals are generally used for this cancer research, etc. and that animal, you can see that animal, it will be like, it does not have any fur, it will be skin, like skin like mice, which is called as a nude mice. So that nude mice is an example of a transgenic animal which is created only for experimentation purpose. uh, the example nude mice is the one where the animal is immunocompromised, in the sense that it does not have any immune system in its body. So that is with the recommendation. Only such animals can be imported into the country. Next is, it also give training to scientific and non-scientific persons on alternatives, and also how laboratory animal welfare, and if alternatives for animals can be used, what are the alternatives, etc. Such trainings are also given by CPCSEA. So these are the main activities of CPCSEA. Next we will see what is, as I said in the previous slides, an animal ethics committee. An animal ethics committee, as I said earlier, it is an institutional level committee where the head of the institution is responsible for maintaining the animals, and is responsible for constituting this animal ethics committee. So animal ethics committee ensures that the experimental conducted are rational or are important. No unimportant projects will be sanctioned with animals, and that experiments does not cause, or those do not cause undue pain or suffering to the animal, and only minimum number of animals are used. So this is controlled by the animal ethics committee, which is an institutional level committee constituted by the head of the institute. So this institutional animal ethics committee, which is IAECS, approves the research projects or proposals that uses rats, mice, guinea pigs, rabbits, etc. So the guidelines of laboratory animal care and use are formulated and is enforced by CPCSEA. So this animal ethics committee, they work with the guidelines of CPCSEA, or they are under the observation or control of CPCSEA. They are bound to follow these rules. So next is the international animal protection initiatives, which can be related to this one, which is called as a concept of 3 Rs. So whenever an animal experiment is being formulated, they should follow the concept of, or they should keep in mind the concept of 3 Rs. One is refinement, the second R is reduction, the third is replacement. So first one is refinement. So refinement is modification of any procedure that minimizes stress and pain of experimental animals. So you need to refine the experiment. Okay, if some additions are to be done, or you have to remodel the experiment in such a way that it should not cause, or it should cause only minimum stress and pain to the experimental animal. That is refinement of an experiment. Second one is reduction. Reduction is the one that is also enforced by the ethics committee and also by CPCSEA. That is, only minimum number of animals should be used. Minimum number means statistically significant minimum number of animals should be used for the experiment. The third one is a replacement. That is, if possible, animals can be replaced by animal cell cultures or in silico models. So we'll discuss this in detail. That is a replacement. How will you replace animals? So replacing animals, when you start an experiment, directly going on to animals, and now this animal ethics committee will not give you permission. So if you have formulated a drug, okay, from an Ayurvedic extract, you have formulated a drug. For example, you have formulated a drug compound which is and which is having an anti-cancer, which you believe to be having an anti-cancer property. So the ethics committee will not permit you, because you have referred the text, Ayurvedic text, etc. You have done some experiments in the lab, etc. And from the text reference, you know that this is having some anti-cancer property. this particular plant extract is having some anti-cancer property. So you cannot go and directly do an animal experiment. Before doing an animal experiment, you should prove that this is having an anti-cancer property on animal cell lines or cancer cell lines. So cell lines are nothing but in vitro cells. That means usually cells are seen inside the body, which is called as in vivo. When you culture cells outside the body, that is called as in vitro. Okay, so cancer cells have the ability to survive outside if you provide the required nutrition, medium, etc. They can be cultured on a solid substrate. So such cell cultures may be utilized before using an animal. You can prove that if those cancer cells they are being, or if your drug is showing an anti-cancer property in those animal cell lines or cancer cell lines, after those experiments or after validating such experiments, so there you are not killing any animal, you are taking cells from animal cell cultures or cancer cells or cancer cell lines. So from where do you get this cancer cell lines? Cancer cell lines are available in every from every cancer labs. That means if you take from the a live cancer also, such cells can be grown in a laboratory. Okay, because of the or the because of the division, or because of the repetitive divisions of the cancer cells, or they divide or continuously unstoppable division of the cancer cell lines or cancer cells, they can be cultured inside a lab, like inside a test tube, etc. So first you have to test your drug in such cell lines, whether those cells, you can observe those cells in the microscope, you can see when the drug is being added, whether the cells are dying off, etc. After you prove that this particular cell line or cancer cell lines are affected by your drug, you can go to the for animal experiments. So that is by replacing with animal cell cultures. In the next case is in silico. So now we came across two words: in vivo, which means inside the body; in vitro means outside the body. Some experiments or whatever it is, if you are doing outside the body, that is called as in vitro. The third term is in silico. That means if you are replicating this particular experiment on your computer, like you are modeling your drug, you are checking your drug interaction with a particular receptor, etc. Such things are usually referred to as in silico. That means something, if your experiment is done on a computer, okay, with the help of modeling softwares, that is what is called as in silico. So now we came across three new words: in vivo, which means inside the body; in vitro means it is outside the body; in silico means something which is done with the computer. So this is with the animal initiatives or animal protection initiatives. And next is the ethical guidelines for the use of animals in scientific research. So this is just a summing up of the CPCSEA. So the animal experiment should be undertaken only after due consideration of the relevance of the particular experiment. Should be undertaken only with the relevance of that experiment on, or it should have some or that particular experiment should provide some knowledge in health or advancement of knowledge, either in a health sector or advancement of knowledge. The animal selected should be appropriate species, the number, and in that case the minimum number should be used to obtain scientifically and statistically valid result. That's why we always said that minimum number should be used, should treat the animal with kindness, and take proper care by avoiding or minimizing pain or stress to the animal. Provide best possible living conditions or supervision of a veterinary specialist. Use in vitro systems or in silico methods whenever possible. So this is a summing up of the guidelines of CPCSEA. This is considered as the ethical guidelines for the use of animals in scientific research. So next is some animal protection initiatives, where we'll discuss these two, which is Animal Welfare Board of India and Society for the Prevention of Cruelty to Animals, etc., that is SPCA. We'll discuss both this in the next session. Thanks for hearing.